Hi there, Falcons. Welcome to another DI or direct instruction. It's nothing but a preview to the dissection labs happening in room 311. And we can do this every Friday, or we can have something like dissection any days, so long as there will be adults to help me out in the lab. And that would be the teacher candidates from University of Manitoba or University of Winnipeg. Thank you. And of course, there's our educational assistants. And from time to time, we can invite teachers, students, the EAs from the other programs or from other homerooms. And it's all part of inclusion, right? So, and we're happy to share that we now have established a learning partner in Maple Leaf, right? So, this is a very big food company in Winnipeg, Manitoba, or in Canada. And an interesting thing about it is the company is now carbon neutral, right? Mm, big words there. Carbon neutral. What does it mean? So we all know very well that everyone has a carbon footprint, right? From turning on the lights at home to driving to work, we all use energy. And right now, that energy puts more carbon into the atmosphere than it can handle, contributing to climate change through human activity. As our planet gradually warms, that leads to environmental issues we can feel right here in Canada, as well as around the world. That's why Maple Leaf is the first major food company in the world to become carbon neutral, using science based targets to reduce emissions as quickly as possible. Now, how terrific is that, right? Working with Maple Leaf, right? And we're happy that we have established a partnership with them so that we can get for free some of the specimens that we will be using in our lab dissections. So thank you, Maple Leaf. Now, why dissection? So hopefully we can go heavy with dissection this year and in the years to come. Because for me personally, and for you, I'm sure, if there's something as hands on as an activity, it would be dissection, right? So, dissection, the word comes from the Latin word dissecare or dissecare. It means to cut to pieces, also called anatomization, right? Is the dismembering of the body of a deceased animal or plant to study its structure or its anatomy or anatomical structure. Big word there, right? And in autopsy, for instance, um, it's used in pathology and forensic medicine to determine the cause of death in humans or in animals. Now, pathology in particular is the study of the causes and effects of disease or injury. So you can kind of see the real world connection now between doing a dissection in the lab. Maybe in your future work someday, you can be in a pathology department. You're trying to kind of get into the heart of the matter, like what's happening, what's the disease, what's the body part that really causes maybe the death of a person, right? Or if you're working in veterinary clinic, that could also be very helpful. Trying to just get to know what's happening with the organs after death, right? And discovering and uncovering maybe some solutions or some issues on the causes of death, right? So, and of course, with Maple Leaf now, we would be getting some samples from them. Like this is now the kidney. Hopefully, we could just try and compare how it looks like with the diseased kidney and compare it with a normal kidney. And we would be having eye specimens, the cow's eyes or sheep's eyes, and of course the anatomy or the structure of the eye. We can do that. We can dissect what is inside once we started to cut it off or do a cross section of it. And then of course the liver, and then healthy liver versus damaged liver, and the heart, of course, healthy heart versus the damaged heart, and all the underlying issues, problems um, that's causing diseases or abnormalities in all of these organs. We can just discuss it in class, right? So, people, I ask you now the big question: are you ready? Now, of course, we do not just um, hand out the specimen and lay it out on the desk and simply cut it for no reason at all, right? There should be a purpose. There should be some pre-teaching of expected behaviors and the procedures also. So there you go. Now, first things first, you need to clear your desk. And if you have to, before coming into the room, do a washroom break right away, right? So we should clear our desk, and if we can have this, that's perfect. Or if you're just wearing glasses in lieu of goggles, I may have a few in the room. So some of you can just borrow it, right? And I made sure that everything is sanitized. Now, or if you have some clear glasses at home or a sunglass, clear sunglass maybe, you can bring it to class and use it. And the magnifying glass, I do have that in the room, so you can look deeply into what you have cut. So, and this is now the dissection tray. So you can think of it like a metal tray, rectangular tray with kind of like asphalt-looking substance, right? So that the specimen can be placed there, and it could absorb some of the water content of the specimen as we start to cut it up. And of course, sanitizers, right? So before you put on your gloves, for instance, right? So you kind of do this and then put on your gloves. So, and I know the others might not uh, be up to the challenge of doing the dissection. That's all right. So you can just step back and watch your classmates do it on the table. So I'm okay with that. And of course, we can have scissors instead of scalpels for safety purposes. Now, you've seen me wearing at times or most times a face shield like this. You may have face shields at home, so it's also nice to use this to avoid any splattering, just in case when you're trying to cut up the specimen. And then lab coats would be ideal. I think the Inner City Science Center can loan to us some lab coats that you can wear. But if you have a simple polo shirt like what I'm wearing right now, that's fine to bring to the room, and then you can just discard it any. Afterwards, after we're done with the dissection activity. What else do I see here? Of course, in school, we still use the mask, so you can just do this, which is perfect, right? And if you have long hair, like me, you could just bring your own hair ties or elastics and you can just tie up your hair, okay? Like that. So I guess, and then you're ready, right? Or if you're using any, if you have on your person bracelet, you can just remove that, right? So that um, it would not be in, so that it would be more okay to dissect without anything that's kind of bothering you while you're doing the cutting up process, right? So there you go. So and and of course I have here some dissection lab rules, which I'm going to repeat every time we do the lab. So it says there, On dissection day, long hair tied, roll up your sleeves. If you're wearing heavy coats or sweaters, leave that in your lockers, right? And take off rings if you have any rings, right? Sanitize hands, so you know that. And then you will note that in room 311 there will be black garbage bags on the sides. So simply dump the dissected specimen or organs there, right? And dissection tools. So trays, scissors, pins. I will have a tub with pine sol and water, and then I would make a clear note there. Used dissection tools here. So just dump it there so that we can sanitize it for the next class, right? And then, of course, take care with any sharp tools that we are using. Please do not poke your classmates with it or unnecessarily just stop the dissecting tray. Don't do that. And of course, observe careful hygiene precautions after the dissection. So there's a reason why we have the sinks in the room. So kind of wash your hands with soap and water and then dry it up with the brown towels there. Okay, there you go. We're almost ready, right? But in a typical dissection procedure, I wish we could have all of this. But not all of them, or maybe some of them, or some substitutions allowed, right? Instead of a dissecting knife, maybe I could just have kitchen knives there. But in any case, just be careful with it. And I would always kind of demo it. So there will be a next video with Miss Valerie showing how everything is cut up, right? So that even those students who are not with us can follow through and we'll have that virtual experience of dissecting, right? And we will go with a number of dissection labs here. So we can have kidney dissection, uh, eye dissection, liver dissection, heart dissection. So if we could just stage one or two in a month, that would be perfect. And of course, everything boils down to the freshness, more or less, of our orders from Maple Leaf. So allow me to share with you the dissection plans. So you will note that we would be getting a number of specimens. So 25 eyes, 65 kidneys, 30 livers on November 4th. And of course, I will try and sort them out. And it's best if we can do the dissection right away or in the next coming days after we've gotten hold of the orders. And you can see that November, December, January, and Feb, we have a number of specimens or organs coming from Maple Leaf. Now, of course, with this, we can now proceed with the next plan here, and you get to see that. So let me just hide this so that I can see what I have. So with the first lab that we're doing, it's going to be on the kidney dissection. And we have what we call this pre-lab, and then during lab, and then post-lab. So pre-lab is that part, maybe a few days before the dissection day. That's the time when the teacher would be pre-teaching responsible lab behavior. And of course, the dissection rules will be revisited with you guys. And then there would be some pre-teaching on the excretory or urinary system because that's where you can see the kidney, right? And if I'm going to kind of click on this, try and see what's happening here. So the excretory or the urinary system. So how does the urinary system work, right? So its main function, it says here, is to filter blood and create urine as a waste byproduct. The organs of the urinary or the excretory system include the kidneys, the renal pelvis, ureters, bladder, and urethra. Now, this is a front view of the urinary tract. But of course, this is what we are interested in, the parts of the kidney or the anatomy of the kidney. So, how do we get this structure seen by you guys? So, we cut it off in a cross sectional manner or sideways, and hopefully, we're able to see all these different parts. So, big words here: calyces, the renal artery, renal vein, the ureter, cortex, medulla, renal pelvis. So, and of course, we have two kidneys, right? But we could function with one, why not? But then more than one is better, right? And I think that's the explanation, what, like the classic question. If you can function with one, why do we have two kidneys? Because not all are fortunate, right? And would you believe there are even some people born with just one kidney or no kidney, but this would be in extreme cases, right? But in any case, this pair of purplish brown organs is located below the ribs. Where's the ribs? Somewhere here towards the middle of the back. Their function is remove waste products and drugs from the body, balance the body's fluids, release hormones to regulate blood pressure, and control production of red blood cells. So that's part of the pre-teaching done by the teacher, right? And of course, there's a virtual lab that one can go into in case you miss the dissection, and I can just release this side. But I know students are very enterprising. I write a lot of essays. Uh, they're now enterprising, and they could easily just get into the heart of the matter, right? And you can just Google on just anything right now. I also write sales descriptions for ads. And then usually there's the. Let me just skip the ads there, right? And this is from the UK, and you get to see the person just going through the motion of cutting up the liver. So I'm going to do a similar video like this at some point, and so that you can follow through, you can pause, and for those who are at home, they get to have a sense of what's happening, right? So there would be the mass of fatty substances here, right? This one, and you have to clean out the kidney so that it can be revealed. So you have to remove this cautiously till you are presented with the pair of the kidneys, right? So do not totally cut off this part here, right? So, and it would explain to you later on. We would just discuss this in class, like what is this part, right? And then when you cut it off, so sideways again, or this is what we call the cross section, right? There you go. So a pair like kidney beans, right? So, and then that's where you would start to kind of dissect and identify the parts of the kidney. And notice this is the dissection tray. And later on, sometimes it's difficult to identify, or uh, they can be moving, or that's why you can place pins with different colored heads so that we can identify it on the screen, right? What is it, and so on. And hopefully, we can have something like this with colored pins. And if I can project this on the overhead, that would be awesome, right? So that you get a sense of what it is, right? So this is just a preview. And of course, We will brainstorm some essential questions to the science reflection sheet, and this is still part of the pre-lab. And of course, the tick is shared with you, and I'm sure you know what tick means. T stands for task. It answers the question, what are we doing? Clearly, we're doing a kidney dissection. Intent, why are we doing this? It says here, kidney dissections are conducted to explore the function of a kidney balance in the body by examining the external and internal structures. So the kidney filters out metabolic wastes such as urea and some salts and regulates the water and salt. Big words here, urea, salts, right? And it says here, being introduced to new words. So example here would be nephrons and the other parts of the kidney and their functions. And another intent, right? So again, intent answers the question, why are we doing this? So that we could better correlate the actual dissection with the virtual lab. So compare and contrast. Did you miss out anything while you were doing the actual lab compared to what you've seen in the virtual lab? So that's part of the intent. And of course, criteria answers the question, how do we do it? How do we demonstrate that we've learned something from the task or activity? So this is now part of our post lab. So it can be done the next day or the next meeting. So completed science reflection sheet. And this is something now that I can post in Google Classroom so that you can fill it out. So let's just take a good look at preview this with you guys. And then you can always go back to this video later on so that you are reminded of what we have discussed or what I have mentioned in this video. So this is now your I'm looking now at my focus. There you go. So this is now your the student copy of the science reflection sheet. So it would look like this. So the first task would be I can do this, right? I can label now the parts of the kidney after doing the dissection. So there you go. What is this part? What is A here? What is B, C, D, E, F, G? And of course, you can type out, start to just put in the answers, or you can reference again the answers from this video because um, earlier I think I mentioned the anatomy or the parts of the kidney. So you can go back to that part in the video, right? And then part B, lessons learned. Use your own words. Now, it is tempting really for you to just Google for the answers, but we encourage you to use your own words, right? So lessons learned. It's just not as if you just write down what you think you have learned, right? As teachers, I think I find it best to give you guiding questions. So I kind of put in brackets the essential questions, right? So part of your lessons learned would be this: why is the kidney important, right? And another lesson learned can be this. So think in your head: why do I have a pair of kidneys, right? Maybe you heard me talk or reference to that at some point in the video. Go back to that, or you can Google on it. Why not? But use your own words. And another lesson learned can be this. Again, think of these questions in your head. What is a common problem with the kidney? What are the causes? How can it be prevented? And then, of course, the last would be, I like the kidney dissection because, and then you can give me positive comments so that I can also share that positive feedback to Maple Leaf so that we can continue the partnership, right? So there you go, people. So I hope that with our first kidney dissection, it will go as smoothly as possible. Okay, there you go, Falcons. Ciao for now.